My name is Terrence Barkin, and I'm the Executive Director of the Graphene Council, the largest community in the world for graphene producers, researchers, application developers, and end users. We have hosted the Graphene in Healthcare and Medical Applications conference series, and the video you are about to watch is from that series. And I'm really pleased to introduce Vincent Bouchiat, who is the CEO, founder of a company called Grapheal. And you see the title slide, using graphene for continuous monitoring of wounds and healing. And with that, Vincent, I'm going to turn it over to you. It's all yours. Tell us about your company and your applications.
Thank you, Terrence, for this opportunity to present our company. So I would like today to present you the potential application of graphene for wound care. And not only speaking about the product we are trying to put on the market, but also about the translational research that make this possible. We started this project at CNRS in Grenoble in France, which was my hosting laboratory. I'm coming from academia. And it has been an 8 years journey, which is still not finished. And I would like to show you how we came to use graphene for bandage. So as we know, these last 2 days, we had a wonderful set of presentation about the biological and biomedical application of graphene. And we see that graphene can indeed be used for a lot of potential application, even more that there is on this slide, actually, which is already quite old. But I would like to focus on two applications, two possibilities, biosensing and tissue engineering, and potentially mix the two in the same product. First, I would like to tell you that graphene exists in so many flavors, so it's a bit confusing. And I would like to try to precise a bit the terminology, what I consider as graphene in my product. It doesn't mean that other type of graphene cannot be useful for healthcare. As we see, there are many other possibilities. But in our case, we really focus on a very specific type of graphene. So in that graph, coming from this publication, we've seen that there is a possibility to classify and to make some better ranking of graphene depending on the number of layers on the z-axis, on the amount of oxygen grafted, because many graphene come from reduced graphene oxide, and also the size of the sheet, here starting from 1 nanometer to a much longer size. And you see that in this three-dimensional space, there are already very different potential application and potential properties of graphene. In that, for example, you have graphene quantum dots that can be very useful to making labeling of cells, for example, due to the optical activities. In our case, we are talking of graphene of very large size, one monolayer and without oxygen. So it's on this axis, very far away out of this graph, because we are dealing with large crystal of graphene produced by CVD of graphene on copper, and which are transferred on a polycrystalline element on polymers. I'm going to speak a bit how we make that. So microscale monolayer graphene is extremely different for graphene platelets and graphene flakes. So that's no surprise that the properties and the biochemical and the pharmaceutical property of the graphene in that case are very different from the other ones. So nevertheless, there is potentiality of decomposition, degradation of graphene in the body. And in that case, it couldn't come back inside this space, even though it starts from that. So it's very important to know about the decomposition and the byproduct of graphene and the potential toxicity of it. So in the rest of my talk, I mostly speak about this type of graphene, which is a mosaic of polycrystalline graphene, even though here it appears like a single crystal, but it's a monolayer on top of a polymer, which is biocompatible. In the beginning, we decided to use monolayer graphene as a substrate, and we started by making growth of neurons on graphene to try to record the neuronal activities. So that was a very fundamental project in the beginning. And the nice thing with graphene is that it's a transparent layer, so you can use it as a substrate for growth, but you can see through with an inverted microscope, which is, of course, a very useful tool for the biologist, and use the top part to inject fluids. and the in-plane to measure activity. So it's an extremely convenient setup you cannot make with silicon, for example, or with metallic layers. So that's why we use that in the beginning, and we published that in 2018. Interestingly, we found that we can detect a signal and that we could measure action potential and even intracellular potential due to the very specific interaction of pods of neurons with graphene. And during that study, we found that graphene was indeed promoting the growth instead of just letting the growth. So we see that the neurite length was higher than expected and higher than other materials. And not only the neurite length, but also the normalized cell density. So we got not only biocompatibility, but biostimulation by graphene. And the reason for that was not very clear, but one possibility is that graphene is a conductor, so it promotes interaction between cells, but also something very important is that the binding strength of graphene, so the nanomechanical property of graphene exactly match the binding of a phospholipidic bilayer, which forms the cell's side walls. So this matching of properties is actually very important and probably one of the reasons of the biostimulation of the high biocompatibility of graphene. As you can see here, this is the border between coating-free glass and graphene, and you see that the neurites like to follow the border but not interact with the coating-free glass, like if they prefer to stay on the graphene layers. And you clearly see that, for example, on this graph, this is silica and this is monolayer graphene on silica. There have been a lot of potential explanation of that. And in this paper here, you can see another possibility of this biomechanical matching, which is proposed in this paper about this way graphene is promoting proliferation and spreading of living cells. So then we decided to look at skin cell culture. This is the human fibroblast. And we mimic a wound here by scratching the layer without scratching the graphene. And you see that on the control substrate, after 14 hours, you still have a gap here. It's a ½ millimeter gap. In the case of monolayer graphene coated slide, you clearly see that there is the cells at the center and there is full proliferation of the system. So, and a higher density in the beginning. So again, we reproduce that with skin cells. And then we went to in vivo, and in that case, we use a diabetic mouse, which has a delayed wound healing. And indeed, after 12 days, you have a smaller wound when you have this polymer coated with monolayer graphene compared to the control one with the same polymer without graphene. And we did that also with pig model, which has a skin extremely close to the human skin. So this speed up, it goes in the very few days here. If you look at the side, instead of having extension of the wound by inflammatory response and then coming down, you clearly see that there is a closing of the wound on the first few days. And this time here gained 3 days, typically on the mice wound healing. In the case of pigs, it's a bit less prominent, but you have still typically 15% healing speed. In the case of this human fibroblast on substrate, you have also the same similar effect. So this bioacceptance can be also very useful for implants. And this is, for example, an intracortical implant for probing the activity of a mice brain. And this is the control with a commercial implant. And the same implant coated with graphene, and the red elements here, which are stained astrocyte, which has the inflammatory response of the hallmark of the inflammatory response of the brain, are much smaller in the case of coated graphene. That's quite important because it suggests long-term acceptance of graphene. And if you look here, for example, in time in weeks of the measurement of this probe here, taken in situ, you have a much longer signal. You keep it much longer, up to 5 weeks, compared to the commercial implants. And these graphene-coated implants could have a very short-term application here. So how can we make this for wound healing? Instead of coating silicone or hard sample, we decided to make a transfer on polymer. So we start from graphene, monolayer graphene on copper, like it has been shown yesterday. We deposit a supporting layer of a biocompatible polymer, typically 5 to 10 microns thick. And then we etch back the copper foil and we remove all copper ions. That's very important. And you end up by putting upside down the layer with a graphene exposed and backed with biocompatible polymers. And this we insert it in a typically bandage. And this we can apply on wounds. That is now possible at low cost because, as we've seen now, there is a lot of producer of graphene on copper. We've seen yesterday the presentation of Mr. Drab from Gore-Tex, but there are many others. And what's very important is that this large-scale production makes the product and the cost of this bandage much more acceptable for a consumable product, because now it's much less than $1 per centimeter square, which extremely make it feasible in terms of economical solution. So why graphene on polymer is an ideal material for making bandages? First, we've seen it's biostimulating. It's a conducting material, so you can use as an electrode to apply voltages, and that's quite important because there is a large field of research and even product on the market where you electrostimulate the wound by application of pulses of voltage. It's also transparent, so you can see through, which is quite important for a bandage. Extremely flexible because we can make film of 5 micrometer, which are very conformal and it can go into deep wounds. And it's sensitive, sensitive in the means that you can measure the resistivity side to side of the bandage, and this will give you an idea of the composition of the liquid in contact with the wound. So importantly, we have to check that the graphene on this material is stay integral, it's not leaching out nanoparticle. Here you see two same micrographs taken on different places of the same bandage I've shown you before on a pig after 3 days. The scale of the graph here is about 20 microns. You clearly see that there is indeed a little bit of defect, starting of delamination because of the rubbing effect of the wound on the graphene, but very little amount are actually being released. It's less than 5% and it's a monolayer, so the quantity leaching out is extremely small, and actually it's probable that there is delamination but still no leaching of nanoparticles. But we can quantify that very importantly quite precisely because we know that there is only one monolayer on the material at the beginning. So we checked also the non-toxicity of these materials, so by using typical elements for pharmaceutical compounds. So there is a well-defined ISO standard for this kind of element in contact with the wound. And this was passed all together. So we ended up making a product out of it. So why graphene can be useful for wound care? Well, it's biocompatible. It's also biostimulating slightly. You can apply voltages, but there are other things you can do with it. So we wound up really making a bioelectronic sensing platform, by the way, not only for wound healing, but also to measure saliva compound and other type of biomarkers. So we combine for that 3 technology, which are actually quite mature, this graphene bioelectronics, which is now scalable, which you can use both as a healing platform, but a sensing one at the same time. We couple it directly to a smartphone, and that's quite important because smartphone is the basic tool for all nurses, especially the nurse that goes on the field. And for that we use antenna and a small chip to record the activity and the evolution of the bandage. And finally, because of the smartphone gather the information, you can couple that to the telehealth platform, which developed very fast, in particular for diabetic people. So chronic wounds is a big problem, which an untapped potential, because there is no such thing on the market as a smart bandage yet, even though there are a lot of academic studies that are quite promising using graphene, but other material as well. It's a growing global problem because of the 2 reasons. One, because there is an increase of elderly population. People are aging fast in many countries. And also because there is diabetics importantly, which rise. And during the disease, 25% of the diabetic will get foot ulcers. And foot ulcers are deep wounds that can end up in 20% in amputation. It corresponds to 500,000 amputation per year worldwide. It's a very important problem in US, for example, which costs billions of dollars for the healthcare systems. It's always important because, you know, the wound is below very large bandages, compressive one, and there is a lack of information. And even the nurse lack information about the follow-up of these wounds. And it stays during months and even years sometimes. So it's always important to keep the information because of the turnover of the caregivers. So digital solutions are definitely needed in that case. So that's why we propose this kind of bandage to record and make a journal of the wound. So the bandage will have dispatch that will be kept, and of course is a disposable product. So when we change the bandage, we change the patch as well. This patch will be used for monitoring first, but also for electrostimulation and as well as a biostimulating material. So the idea of a combined therapeutics with diagnostic is really the added value of graphene in this kind of bandages. So the thing is that you have to put this on the side of the wound to measure the liquid exudate. So there is an important problem of having this exudate that goes on the sensors and at the same time part of the graphene in contact with the tissue which proliferates. So the important also is to make this working on the foot. So there are a lot of challenges because of all the mechanical problem created by that. The elements here need to be integrated. So you see the graphene part of the biosensor, some electrodes, a chip, and the antenna, which we connect with the smartphone, directly with the smartphone without the need of a specific readers. So what we measure first is directly without making any functionalization of graphene. Graphene is sensitive to the ionic force, the amount of salt, but also to the pH. And you have a significant variation of the resistance of typically the bandage as a function of the variation of physiological pH. There have been propositions in the literature to make also detectors of other type of pathogen, like here in this work here of Staphylococcus, for example. Importantly, we have on polymers good reproducibility of devices which operate like a field effect transistor, like we saw yesterday in a biosensor for point of care. So how the system is working? So this patch is making a continuous monitoring. It will detect infection at early stage when there is an infection starting to take in a wound to have a rapid change of pH, but also of temperatures. And the chip can measure variation of temperature as well locally. And these two parameters acquired simultaneously will be sent to the smartphone and with a possibility to be analyzed by the remote systems, which can send back information to the nurse. So that is a bit where we are. As a conclusion, what we can have here with graphene as a versatile material for bioapplication. It can make sensitive biosensors. You can make biosensors targeting different type of biomarkers, even though you have to redo the biocompatibility each time you put a different bio probe. It's a SATO-compatible material, which lowers long-term inflammation. It can actually use as a diffusion barrier and metallic conducting elements. And it can be a important part for soft electronics using printed electronics. You can make it at rather low cost with a comfortable application. So I finish here and I open for questions.
Vincent, thank you so much. That's impressive amount of work. When did you start Grapheal? How long has this been in process?
So the company has been incorporated in 2019. So we are a bit more than 2 years down the line. And we are focusing on this connectivity plus graphene sensors and with the wound care focus.
Well,
But
that is an awful lot of progress that you've made in a very, very short period of time. I'm curious with the work that you've done, what surprised you the most out of what you've done? What did you not expect that you discovered through this process?
well, there was first a gamble about the fact that graphene will become a commodity with sufficiently low cost so we can make it a consumable product. And that was the big surprise, is that you can make now square meters of graphene per hours with the same quality as the one you used to have with Scotch tape, almost. So there are, of course, polycrystalline system, but it's getting better day after day. And so that is for us the biggest surprise and the nice surprise that makes this possible. So that's, I would say, the evolution of the graphene industry that can support such applications.
And what's the biggest technical challenge you face at this point, or do you feel like you've addressed most of the technical challenges? Now it's more a business case.
Actually, there are many other technical challenges. You see, it looks simple, but there are microfluidics, the anti-fouling of graphene to prevent the protein to adhere on graphene that will actually shield the sensors. There is a lot of technical hurdle. However, I think we can solve that. And the thing is also is to define something which we can call medical grade graphene, which has this quality enough to make it as a material in contact with open wound. And that is something to define with the standards, which are required for pharmaceutical compound. And that also is a big challenge, even though we have good hope we can make that very soon.
Well, maybe that's a discussion we need to have, because the Graphene Council is about to publish our graphene material classification framework. And
that's extremely important.
It would be possible then to use the characteristics and the value ranges within that to define, the type of material that's suitable for this given application. That's one of the reasons we designed it, is to help with that kind of a problem. So it'd be great to have your input on that. This application for healing, and very well aware of the problem with the diabetic condition and with chronic wounds, which, people really need to understand. These are open wounds that never heal because of an underlying medical condition and constantly need to be monitored and managed. Have you looked at other applications, for example, treatment of burns and things like that, or are you really focused on just primarily the diabetic wound care?
So diabetic wound care is a well-established condition with, unfortunately, a lot of people affected. So it's a very clear business case in terms of economics. Burns are also extremely promising, of course. However, in terms of clinical study, burn is very much more intimidating because burns happens after an accident. There are never two burns the same. There are two diabetic foot ulcers the same, you see. So right. For making clinical studies, burns is extremely difficult, even though it's fascinating, of course. We are also looking at surgical wounds and the possibility to actually monitor during the first few days after surgery, the fact that there is no, for bariatric surgery, for example, no infection starting.
Yeah, I think the possibilities are really quite broad. I was fascinated when you said that the material had a biostimulation effect. That's not something I had expected.
For us too, it was a big surprise that actually triggered this company.
Yeah, that's fantastic. We have some questions from the audience, and this comes up quite often. I think we know the answer, but please let us know. One of the questions is there any problem associated with cytotoxicity of graphene when in direct contact next to human skin? And I know that there have been at least a few studies about dermal contact, but talk to us about what the cytotoxicity effect is of graphene in direct contact with the skin.
Yeah, I think this is a very important question, and we cannot overlook that. It's a very central point here. And I receive a lot every day question on my mail about that. as you know, even in Europe, there is this conspiration theory about graphene everywhere in the vaccine and so on. So we are on the front row with that. So I can tell you firsthand that this is a very, very tricky question. We've done our work with our material. So it's all about the material. And as you know, as I said, so many different graphene. So there are graphene which are cytotoxic, probably because of the edge, probably because also of the oxide. In our case, which is without edges or very minimal amount of edges, and because it's a continuous layer, and also because there is no oxide, I believe that our material, and we have, not only a belief but proof that it's rather low to negligible cytotoxicity. Mm-hmm. There have been a lot of studies with graphene, and I can show you that one about the degradation of graphene also. This work here is a follow-up of graphene, pure graphene in vivo in mice, in lungs, for example, and they show indeed that there is biodegradation of the crystalline graphene into amorphous carbon, which is also very cytocompatible. So, of course, there are questions, and we need to be open to any problem and to have this vigilance. But I would say, the amount of nanostructure of carbon I put in my bandage is less than the diesel fume you have when you cross the street. Huh? No, that's the fact. It's not. To give perspective about what we put. Huh?
Absolutely, I think you're absolutely correct. We need to put this in the correct context and deal with facts. And if there are issues, then we need to address them, because there are many other substances we use every day that have much higher problematic issues. We just simply understand how to use them correctly. I want to get a couple more questions in before our time ends here at the half hour. One question relates to where this can be used. Could it be used, for example, on tissue like the eye?
Probably. This is something we got contact with ophthalmologic about that. I cannot give any feedback yet. It's too early to say. But indeed, graphene has been proposed also for smart contact lenses, as a monitoring of glucose, for example. So there are plenty of possibilities. Since graphene now is cytocompatible in some points, at least this one, and it's also a sensor. So potentially, yes, it could be extremely useful for eye.
Let's try to get just two more questions in here. Another of our audience is asking, why not just incorporate the graphene directly into a polymer and make kind of like a composite, as opposed to a CVD process where it's a monolayer and then it's added and adhered to it? What would be the reasoning?
Extremely good question. In our case, we would like to have a conducting layer on the macro scale. I measure the graphene side to side, centimeters away sometimes. So you cannot do that with a polymer. This is the first question. The second one is, in a polymer which is loaded with graphene, you have much more edges and nanoparticle leaching out. So in terms of cytotoxicity, it's another problem. The dose is a poison, like we say. So here, the amount of graphene is presented to the wound, but in terms of volume, that can potentially be leaching out in the body, it's completely negligible. It's about less than nanogram per centimeter square.
And well, that also speaks to another question, was about the transparency of the material. A monolayer, a single atomic layer of carbon, sp2 bonded carbon, is, I believe, 97.2% transparent. Right? It's 2.7% non-transparent.
Yeah, it's very slightly gray. So you can see it by the bare eyes. So you see graphene with respect to no graphene on the bandage as a small, smoky contrast, but it's very, very transparent indeed.
And one more question was asking, how is it possible for the graphene itself to detect pH, for example? So if I understood your presentation correctly, there's not another bioactive agent on the graphene. You're using the graphene and resistivity itself in those signals to measure. Is that correct? And how does that work for detecting pH? And I think you had some other parameters that you were testing.
Yes. So if you want to, graphene is is open to the system, so it's absolutely not targeting it's just a sensor of charges, of ions. Okay, so any change of ions composition, any charge on the surface will be sensed. Okay, so that's both an advantage and a drawback. If you want to make it specific, you need to graft protein that will target specifically a given biomarker. Okay, without this, the system is sensitive to iron, and so you have basically an iron sensor, okay, which is not selective. But we start with that because this is the biocompatible material. This is the easiest way. We've done all the biocompatibility with that material. So any change of ions composition will be sensed globally. So we have a signal which is not exactly a pH meter, but more like a mixed pH ionic meter. Okay?
Vincent, I have to say thank you at this point because we've come to the end of our time, but I really, really appreciate the information you've shared. I wish you nothing but success with your endeavor here. It's really quite innovative, and thank you for sharing that with our audience today. On behalf of the Graphene Council, I want to thank everybody for participating and for your interest in this sector. Clearly, graphene has a very important future in healthcare applications, whether it be for devices, medical sensing, diagnostics, or even in some of the infrastructure of gowns and devices and elastomer products, etc., that can be enhanced with graphene. So with that, I want to thank you very much. Thank you for your interest in the Graphene Council. If you have any questions, if you need any guidance, if you need information, anything to do with graphene or any of the companies that you saw in the past 2 days, please reach out to us, and thank you for your time and attention.